Welcome to the deep dive. You know, this is where we sift through a whole stack of fascinating sources and we pull out the most important nuggets, the key insights, basically to get you truly well informed and fast. And today, wow, we are plunging into something really monumental. We're talking about a global health challenge that spanned decades, taken millions of lives, really reshaped societies. But this year, it might just be getting, well, maybe quelled by a single incredible breakthrough. Imagine, after 44 years, a recent FDA approval that promises 100% protection against HIV. It almost sounds too good to be true, doesn't it? A scientific triumph that could genuinely rewrite history.
It absolutely does sound almost unbelievable. And the excitement, it's not just hype, it's really deeply rooted in the profound impact this could have.
Right.
What we're talking about here is nothing short of a game changer. This isn't just like another small step forward in HIV prevention.
No.
It really has the potential to fundamentally alter the entire trajectory of the epidemic. It offers a future that, frankly, many people thought was still decades away. Millions of lives could be protected, communities transformed, and that fear, that sort of looming shadow for generations, it could finally start to recede.
So our mission today, then, is to unpack this landmark achievement, really get into all the intricate details. We'll delve into the fascinating science. It's actually quite ingenious behind this breakthrough.
It really is.
We'll trace its incredible journey, from the lab bench to market and explore its profound global implications. But that's definitely not all.
No, we're going broader.
Exactly. Our goal is to give you that truly holistic picture. Humanity's progress, yes, but also, our pitfalls.
Yeah.
So we're going to broaden our lens significantly. We'll also reveal other top scientific breakthroughs of the past year.
Yeah, there's some amazing stuff.
Showcasing the breathtaking breadth of human ingenuity, right?
Right.
Across totally diverse fields, from unraveling the cosmos to rethinking our planet's inner workings.
And crucially, we also need to reflect on some significant breakdowns.
Yes, the challenges in science and global affairs. Moments where we stumbled, maybe where cooperation just faltered, or where, honestly, lessons from the past seem to go completely unheeded.
It's really about understanding that full spectrum, isn't it? Our collective journey as a species. Those moments of incredible triumph where our intellect and determination just shine.
Yeah.
And then the areas where we still face these immense hurdles, often self-imposed hurdles, in translating that knowledge into equitable, sustainable, global progress. It's a compelling look at the current state of our shared human endeavor.
All right, let's dive in then. Let's unpack this monumental leap. We're talking about the dawn of a new era, effective near-total HIV prevention. And it all kicks off with a landmark approval that really sent ripples of hope across the globe.
Indeed. So July 26th, 2025, the U.S. Food and Drug Administration, the FDA, they gave their stamp of, pretty unequivocal approval to a novel drug called Lenacapavir.
Lenacapavir.
You'll see it marketed under the brand name Yeztugo. And the core of its significance, the reason it's being hailed as truly revolutionary, is that it offers this unprecedented level of protection against HIV infection, specifically as pre-exposure prophylaxis, or pre-P.
And here's where it gets really interesting for practical application, where it truly sets itself apart. It's the convenience factor.
Oh, absolutely.
We're talking about just twice yearly injections, twice a year. For anyone familiar with HIV prevention or treatment, that's just a dramatic shift.
Jeez.
Think about what that means compared to the daily pill regimens that have been the standard for pre-P. This isn't just a new drug, it's a transformative pre-PP option. This kind of infrequent dosing could revolutionize uptake, adherence, and ultimately the impact on a global scale.
It is, and this isn't an exaggeration. It is a historic day in the decades-long, really arduous fight against HIV. Daniel O'Day, he's the CEO of Gilead Sciences, the company behind Yeztugo.
Right.
He stated quite directly that this drug offers a very real opportunity to help end the HIV epidemic. And when you look at the initial efficacy data, you start to understand why that statement isn't just hyperbole, why it's, a statement made with genuine evidence-backed hope. The drug provides almost 100% protection for HIV-negative individuals.
Almost 100%.
The pre-exposure prophylaxis trials showed around 99% protection from contracting the virus through sex. And Science Journal, the prestigious academic journal, They went even further. They confirmed 100% efficacy in one trial and an absolutely astounding 99.9% efficacy in another.
So, okay, this isn't just a small improvement, it's a massive leap. But help us understand, why is this particular level of efficacy, that 99.9 or 100%, why is that so profoundly significant? I mean, we've had effective pre-AP before. What does that extra tiny fraction mean in the real world?
That's a really crucial question. And the answer, I think, lies in both the practical and the psychological impact. While the existing pre-P options were definitely powerful, achieving this kind of near certainty in prevention fundamentally removes a major barrier, a barrier that has hindered widespread adoption. For individuals, knowing there's a virtually guaranteed level of protection, that they can truly be, almost bulletproof against infection, they can profoundly impact their willingness to take the drug.
Right, the confidence.
Exactly.
Yeah.
And crucially, their adherence to the regimen. It shifts the whole conversation from powerful protection that reduces risk to near total certainty that eliminates fear.
That's a big shift.
It's huge. And that can drive uptake and consistency in a way that previous options, while effective, sometimes struggled with. It moves from managing risk to almost eliminating it for the individual. That transforms the landscape for public health campaigns. It empowers individuals in their health choices like never before. It tackles that human element of fear and uncertainty head on.
That's a fantastic point about the psychology of it and how that ripples out into public health. Okay, but let's dive into the how. How exactly does Lenacapavir do this? Achieve this incredible, unprecedented feat? What's the core science? The mechanism?
Okay, so this is where the real ingenuity lies. It's a brilliant piece of molecular engineering. Lenacapavir belongs to a new, completely different class of drugs called capsid inhibitor.
Capsid inhibitors, okay.
To really get how it works, let's visualize the HIV type 1, HIV-1 capsid. Imagine it like a tiny, intricate protein shell, almost like a meticulously constructed protective cone.
Right.
This cone is absolutely crucial for the virus's survival and replication. It acts as a secure container, housing and protecting the viral genetic material. And it's essential for transporting the virus with its precious genetic cargo into a host cell.
Like its little suitcase or
maybe more like its incredibly resilient, highly specialized armored car, designed to navigate the harsh environment outside the cell and deliver its destructive payload safely inside.
So it's not just passive storage, it's active transport and invasion.
Exactly. And normally, once that armored car successfully gets inside a host cell, the capsid is designed to shed it. It falls apart, but in a very precise, controlled way. This lets the virus's genetic material get released.
Which is the critical step for it to start copying itself.
That's the critical next step, yeah.
Yeah.
It lets the virus begin copying itself, replicating, basically taking over the host cell's machinery. What Lenacapavir does, quite ingeniously, is fundamentally stop that entire process from happening.
Just jams it up right at the start?
Completely. It's a totally new approach compared to many older HIV drugs that targeted, say, different viral enzymes involved in replication or assembly. This drug hits the virus right where it lives, at its very structural core. It disrupts its ability to even start its life cycle within a cell.
That truly is the why it matters nugget right there, a completely novel pathway of attack. But it's even more fascinating because you mentioned earlier, researchers in the 90s, early 2000s, they didn't even think the capsid was a druggable target. They thought it was too stable, too complex. So What changed? What was the aha moment that completely rewrote the playbook on attacking HIV?
That's a really compelling part of the scientific discovery story. Early researchers, they had this assumption that blocking the capsid's function would require just an overwhelming number of drug molecules, each needing to bind to multiple capsid proteins simultaneously. It just seemed practically impossible to achieve therapeutically.
Right, too many locks to pick at once.
Kind of. The aha moment, the real game changer, came with new, much more detailed findings. They revealed the capsid cone isn't just a simple, rigid, impenetrable structure. It's actually stable, yes, but incredibly flexible. It's a lattice of these 5 and 6 molecule groups.
Flexible.
Yeah, dynamic. It breathes, it changes shape in subtle ways to do its job. This new understanding of its dynamic, flexible nature, its sort of precise molecular dance, that's what intrigued the Gilead chemists and directly led to creating Lenacapavir. They realized they didn't need to use brute force. They needed to understand its subtle mechanics and find a way to jam those specific movements.
So less like a sledgehammer, more like a tiny, precisely placed wrench in the works. And you mentioned a dual action earlier, not just blocking interactions, but actually stiffening the capsid. That sounds like a double whammy for the virus.
Precisely. The research revealed an even deeper, more sophisticated mechanism. It was initially thought the cone just immediately fell apart upon entering a cell, but it turns out it actually stays intact for a while. It acts as a kind of reverse transcriptase container. It even has this astonishing ability to squeeze through tiny pores in the nuclear membrane to deliver its genetic payload right to the cell's control center.
Wow.
So lenacapavir has this remarkable dual action. It doesn't just block the capsid's crucial interactions with cellular proteins, which stops the virus from moving around and signaling effectively inside the cell. It also physically makes the cone rigid.
Ah, okay.
Imagine taking that flexible, dynamic armored car and suddenly turning it into a solid, unyielding block of concrete. This induced rigidity stops it from slipping into the nucleus to deliver its genetic payload.
Completely stuck.
And furthermore, in a really elegant secondary effect, even if some HIV proteins somehow manage to get produced, the drug stiffens the freshly minted capsid subunits. It literally interferes with the proper formation of new viral particles. So it's not just stopping the current infection cycle, it's sabotaging the production of the next generation of faulty, non-infectious armored cars. It's a remarkably comprehensive structural assault.
That is incredibly sophisticated, attacking it multiple ways right at its core structure.
Well-
Okay, and then there was this initial hitch. You mentioned that it was relatively insoluble, hard for the body to absorb if given, say, as a pill. But somehow, that very weakness became its superpower. How did that happen? That sounds counterintuitive.
Yeah, it's one of those beautiful ironies you sometimes see in scientific discovery and drug development. Its poor solubility did mean it would be difficult to administer as a convenient daily pill, which is usually the gold standard for compliance. However, when Gilead's team developed an injectable form of the molecule, this very characteristic, its low solubility and slow degradation, became its superpower. Because it's released so slowly from the injection site over a really extended period, it has an extraordinarily long life in the body.
Ah, so the injection makes the slow absorption a feature, not a bug.
Exactly. That pharmacological property is precisely what allows for that incredibly convenient twice-yearly dosing schedule. It transformed a potential drawback, a chemical limitation, into its most significant practical and public health advantage. It's what makes it revolutionary for adherence. It's a great testament to how overcoming one challenge can unlock unexpected benefits, sometimes in the most surprising ways.
So the science is clearly groundbreaking, highly sophisticated, ingeniously applied. But what about its journey to market and its clinical triumphs? Was lenacapavir always intended as a prevention drug, or did it have a different origin story?
Not initially for prevention, no. That's an important piece of its history. Lenacapavir, under the brand name Lenca, was actually approved back in 2022.
Lenacapavir, okay.
At that time, its primary use was for treating individuals already infected with HIV, particularly as a salvage treatment for those whose virus had developed extensive resistance to other existing drugs.
Ah, so for really tough cases.
Exactly. These were often patients who had pretty much exhausted nearly all other treatment options and were facing really dire prospects. So it already had a proven history of efficacy in a treatment context, offering a lifeline to those with highly resistant HIV strains. This definitely laid the groundwork and built confidence for its potential as a preventative.
That's important context. It wasn't just pulled out of thin air, and it was already getting significant recognition even before this new pre-EP approval, right? I remember Science magazine naming it something pretty significant last year.
Absolutely. The journal Science actually named lenacapavir the breakthrough invention of the year in 2024, which was notably before its official pre-EP approval. They recognized its off-the-chart success, specifically as a pre-exposure prophylaxis, in the trials that were already making huge waves. The excitement was already building based on that preliminary data, which was, as you said, just astounding. That kind of preemptive recognition from such a prestigious scientific publication really underscores the profound impact it was already projected to have.
And that excitement was clearly justified by the clinical trial results that followed. These trials were designed to be robust, right? Diverse populations, different geographical locations. Tell us about those results. What did they show?
The clinical trial results were truly remarkable, almost unprecedented in their clarity and effectiveness. In June, there was an efficacy trial involving over 5,000 African adolescent girls and young women in South Africa and Uganda.
A really key population.
Absolutely. And they reported something that just stunned the scientific community. Not a single person who received the injections became infected. Zero. That's 100% efficacy in a high-risk population. A clear, unequivocal victory. Then, just 3 months later, in September, a second trial conducted across 4 continents, intentionally designed to be even more globally representative.
Okay.
This study involved over 2,000 cisgender men, transgender men and women, and non-binary people who had sex with men. In this incredibly diverse group, there were only 2 infections recorded, which confirmed an astounding 99.9% efficacy.
Wow.
99.9%.
These numbers are simply unheard of in large-scale clinical trials for preventative measures against infectious diseases.
As Mitchell Warren of AVIC put it, you don't see data like this every day. It just speaks to an undeniable, almost perfect effectiveness.
But beyond the sheer numbers, what does this level of protection mean for the practical challenges people face, like adherence, like stigma?
It means everything, really. From an expert perspective, someone like Carlos Del Rio, a distinguished professor at Emory University, he noted that Yeztugo could greatly address key barriers like adherence and stigma. He highlighted how these issues often plagued more frequent pre-P dosing regimens, especially the daily oral pre-PP. Consider the real-world scenarios. For young women in some communities, taking a daily pill might expose them to questions from family or partners, creating stigma.
Yeah, that's a huge barrier.
Huge. And for anyone, just remembering a daily dose can be a challenge amidst busy lives. The research consistently showed that many people who needed or wanted pre-P actually preferred less frequent dosing.
Makes sense.
So the convenience of twice-yearly injections isn't just a minor perk, it's a real-world game-changer for implementation. It fosters better adherence, allows people to maintain their health discreetly and effectively, and removes a huge psychological and social burden.
This sounds incredibly promising, almost like a silver bullet for prevention. But, okay, the major hurdle for any life-saving drug, especially one intended for a global epidemic, is ensuring widespread access and genuine affordability, particularly in resource-limited settings. What moves are being made on that front? How do we ensure this breakthrough isn't just for a privileged few?
You've hit on a critical, absolutely essential point. The best drug in the world is useless if it doesn't reach the people who need it most. And this is where Gilead, the pharmaceutical company, has made some very bold and frankly commendable moves.
Okay.
Recognizing the unprecedented public health imperative, they are waiving royalties and explicitly aiming to provide affordable access globally.
Waiving royalties, that's big.
It is. And to achieve this, they've signed royalty-free licensing agreements with 6 generic manufacturers around the world. These agreements specifically authorize these manufacturers to produce and supply the generic version of the drug worldwide. That dramatically increases the potential for broad accessibility at a much lower cost.
That's incredibly significant. Generic manufacturing is absolutely key, especially for a global health crisis like this. What about access within the U.S. itself? Healthcare costs there can often be prohibitive.
Yeah, so for uninsured individuals in the U.S., they may be able to access the drug free of cost through Gilead's Advancing Access Medication Assistance Program, assuming they meet specific eligibility criteria. But the global partnership is even more expansive and represents a huge commitment. Gilead announced a partnership with the Global Fund to fight AIDS, tuberculosis, and malaria, often just called the Global Fund.
A critical player.
Absolutely. A critical international financing organization. The aim of this partnership is to supply enough doses of Lenacapavir for up to 2 million people over 3 years in global fund-supported countries.
Yeah.
And here's the crucial detail. This is to be done at no profit to the pharmaceutical company.
No profit, wow.
That's a significant departure from typical pharmaceutical pricing models for patented drugs, especially groundbreaking ones.
So they're essentially prioritizing public health over immediate profit in these really high-incidence, resource-limited areas. That's a powerful signal. And for which countries specifically are these efforts focused?
Precisely. And in a further strategic move, these license-free generics will be manufactured for 120 specific high-incidence, resource-limited countries. These are primarily low and lower middle-income nations where the burden of HIV is most severe. Peter Sands, the executive director of the Global Fund, he really emphasized the broader principle at play here. He said, this is not just a scientific breakthrough, it's a game changer for HIV AIDS. And he highlighted the fundamental principle that life-saving innovations must reach those who need them most, whoever they are and wherever they live. It's a clear recognition of that ethical and practical imperative to ensure equitable distribution of scientific progress. This kind of partnership really could set a new standard for how pharmaceutical companies engage in global health, balancing innovation with access.
That's a powerful statement and a genuinely commendable approach. However, even with such bold efforts, are there still significant challenges to access and distribution? Because we know getting life-saving drugs to people, especially in complex, underserved regions, isn't always straightforward. It's not just about price, is it?
You've absolutely hit on a critical point that requires, sober consideration. Despite these really robust efforts, the complexities of global health logistics and national realities remain immense.
Yeah.
For instance, while the U.S. has approved it, regulatory approval for the PEP form of Lenacapavir isn't expected until mid-2025 in some other key regions.
Yeah.
That means a delay in availability for populations that desperately need it now.
Right.
The price for the branded drug in developed markets is still unannounced, although the generic agreements are a hugely positive step for many countries.
Yeah.
However, there's currently no specific discount announced for crucial middle-income countries like Brazil.
Which has a huge epidemic.
Exactly. Brazil has South America's largest number of people living with HIV and could desperately use a drug like this. And beyond pricing and regulatory hurdles, the real-world challenges are just daunting. Resource-strapped governments, already overburdened health systems struggling with basic healthcare provision.
Yeah, the infrastructure.
The infrastructure, social unrest, even basic transportation challenges. Think lack of reliable cold chains or roads can severely hamper delivery and uptake in remote or fragile settings. It's a hugely complex logistical and political puzzle. Reaching millions requires much more than just drug supply. It demands robust infrastructure, trained healthcare workers, deep community engagement, and sustained political will. This is a global health marathon, not a sprint. And while Lenacapavir is a new powerful engine, the road ahead still has many, many obstacles.
So the drug is here, the science is proven, and there are significant, if complex, efforts for global access. But how does this revolutionary development fit into the broader, decades-long fight against HIV? Where have we come from, and where are we still going in this fight?
It's absolutely crucial to remember the historical context to truly appreciate this moment. This is a battle that stretches back more than 4 decades, originating in the early 1980s.
Right.
We've come an unimaginable distance since what were tragically called the bad old days, when HIV diagnosis meant horrific wasting, a brutally decimated immune system, rampant opportunistic infections that slowly consumed the body.
A terrifying time.
Terrifying. And an almost certain early death. It was a diagnosis steeped in fear, stigma, and just overwhelming despair.
And then came the initial breakthroughs, those early glimmers of hope that started to change everything, that truly transformed HIV from a death sentence into something, well, manageable.
Indeed. 1996 was a pivotal, almost miraculous moment. Researchers discovered that powerful cocktails of drugs known as highly active antiretroviral therapy, or HAART, could not just slow, but effectively fully suppress HIV in the body.
Fully suppress.
Yeah. This allowed millions of people living with HIV to recover their immune systems, reclaim their health, and live normal or near normal lifespans. Science recognized this as its breakthrough that very year. It marked a huge turning point. Then, in 2011, another paradigm shift occurred with the treatment as prevention discovery. This proved unequivocally that an individual on effective treatment whose virus was suppressed to undetectable levels could rarely, if ever, transmit the virus to others.
That was massive.
Massive. A revelation so profound that science again named it a breakthrough. As more people gained access to these treatments globally, new infections plummeted from a peak of 2.1 million in 2011 down to 1.3 million last year. Treatment truly became a form of prevention.
So the treatment itself became a remarkably powerful prevention tool, breaking that chain of transmission. And we've had other prevention tools as well, right? Condoms, male circumcision, needle exchanges, education campaigns. What about the previous pharmaceutical PEP options? How did Lenacapavir build on or perhaps surpass those?
Absolutely. The foundational oral PEP using drugs like Truvada was approved in the U.S. back in 2012. It offered powerful protection, no doubt. And in cities like San Francisco, it helped drive new infections to near zero within certain high-risk communities, a real testament to its effectiveness.
Yeah.
However, it faced significant challenges. Years passed before poorer countries could even access generic versions of the drugs, creating a glaring equity gap.
Right, the access issue again.
Exactly. And in many African nations, for example, young girls and women struggled with adherence due to a whole host of complex obstacles, the stigma associated with taking an HIV drug, partner violence, or just the practical challenges of remembering a daily pill in chaotic environments. Then in 2021, cabotegravir was approved, an injectable PPAR-EP that only required shots every 2 months. While that was an advancement in convenience over daily pills, it too was held back by relatively high costs and, perhaps surprisingly, limited interest compared to what we're now seeing with the even less frequent dosing and the global access initiatives planned for Lenacapavir.
So despite all this incredible progress, this whole arsenal of tools, we haven't quite reached our global goals for eradicating new infections, have we? It sounds like we've kind of hit a plateau.
Unfortunately, you're right. Progress has stalled in recent years. The Joint United Nations Program on HIV/AIDS, UNAIDS, they set really ambitious goals, cutting new infections globally to below 370,000 next year and less than 200,000 by 2030.
Ambitious goals.
Very. And we are currently significantly off track for those targets. This is precisely where Lenacapavir could potentially break the impasse. Its unprecedented efficacy, combined with its convenient dosing and planned global access initiatives, could be the catalyst needed to reignite progress and potentially move us much closer to those ambitious numbers.
And maybe even push beyond.
Well, looking ahead, Gilead is already planning trials for an even more convenient one-shot-a-year version, pushing the boundaries of long-acting prevention even further.
Once a year. That would be yet another monumental leap forward in ease of use and public health impact. But Jean Morazzo, she's the director of the U.S. National Institute of Allergy and Infectious Diseases. She pointed out that even Lenacapavir is no substitute for a vaccine. Why is a vaccine still considered the ultimate goal, the sort of holy grail, even with this near-perfect prevention drug? What's the fundamental difference?
That's an incredibly important distinction and a critical long-term perspective. While Lenacapavir is remarkably effective as pre-PP, it still requires ongoing administration, whether it's twice-yearly injections or even a potential annual shot, and consistent engagement with the healthcare system.
Right, you still have to get the shot.
Exactly. A vaccine, on the other hand, fundamentally changes the entire equation. It could potentially be given to everyone universally, not just those identified as high risk. Once administered, it would train an individual's own immune system to recognize and fight the virus,
Creating internal protection.
potentially offering true, durable individual immunity that lasts for many years with perhaps just a few shots. From a public health standpoint, a vaccine would likely cost significantly less to manufacture per dose, maybe only a few dollars, and could reach populations at an even greater scale than a therapeutic intervention. It offers a self-sufficient long-term solution that bypasses the complexities of drug delivery and adherence issues entirely. It would truly prevent infection at a biological level for the masses, offering a pathway to potentially eradicating HIV altogether.
So Lenacapavir is a potent, transformative addition to the arsenal, addressing current challenges in adherence and access head-on. But the search for that widely accessible, universally effective vaccine continues. Overall, though, it sounds like this drug really is poised to accelerate the transformation of HIV/AIDS from a devastating epidemic to a rare, manageable condition.
It is. Lenacapavir is another spectacular biomedical breakthrough in a long, hard-fought series. These breakthroughs are steadily moving HIV/AIDS away from being a disease that upends entire communities and spirals into societal catastrophe, and turning it into something far less devastating, even a rare malady, as it reaches the people who need it most. It's a remarkable testament to sustained scientific effort, demonstrating what's possible when human ingenuity meets a global challenge with persistence and strategic investment.
That's an incredible story of scientific triumph and a profound step forward for global health. But as we said at the top, our deep dive isn't just about the top breakthrough. Science magazine, in its annual review, also highlighted other incredible advancements across diverse fields, really showcasing the breathtaking breadth of human ingenuity in 2024. Let's explore some of these other game-changing innovations, the runners-up that are reshaping our understanding of everything from human biology to the very fabric of the cosmos.
This really broadens our understanding of how science is pushing boundaries in so many different areas at the same time. One significant development recognized this year was unleashing immune cells on autoimmune disease, a revolutionary new application of CAR-T therapy.
CAR-T therapy. Right, I remember that name vividly. It was a huge, groundbreaking breakthrough for blood cancer back in 2013, completely transforming treatment for certain leukemias and lymphomas. So, for our listener, before we dive into its new uses for autoimmune diseases, could you give us a quick refresher? What is CAR-T therapy fundamentally, and how did it revolutionize cancer treatment?
Absolutely. Think of CAR-T therapy as a highly personalized form of cellular immunotherapy. In its cancer application, doctors take a sample of a patient's own T cells. These are the powerful killer cells of our immune system, like highly trained sentries.
Okay.
In the lab, they genetically engineer these T cells to express a special receptor on their surface called a chimeric antigen receptor, or CAR-R. This CAR is designed to specifically recognize and bind to a unique protein found on the surface of cancerous B cells.
Like a targeting system.
Exactly. Once these engineered CAR T cells are infused back into the patient, they act like guided missiles, seeking out and destroying only the cancer cells, leaving healthy cells relatively untouched. This revolutionized treatment for patients with certain blood cancers who had exhausted other options, often leading to dramatic, even curative responses.
That's a powerful tool against cancer. So how is that same principle now making such striking strides against severe autoimmune diseases like lupus, scleroderma, and myositis? It sounds like a natural yet brilliant extension.
It is. The striking improvements seen this year apply that same precise principle to autoimmune diseases. In conditions like lupus, scleroderma, and myositis, the patient's own immune system, specifically certain B cells, mistakenly produces toxic autoantibodies.
Antibodies attacking the self.
Exactly. Antibodies that attack their own healthy tissues and organs. By engineering the CAR T cells to target and relentlessly deplete these problematic B cells, doctors are seeing remarkable results in severely ill patients. It's like a complete system reset, wiping out the source of the autoimmune attack.
So it's taking a tool developed for one devastating disease and repurposing it to fight another equally devastating set of conditions. What kind of success stories are we seeing in patients? What does this mean for their quality of life?
Well, back in February, German researchers reported on 15 patients with lupus, scleroderma, or myositis, which is a debilitating muscle-damaging disease. What they found was truly astonishing. All 8 of the lupus patients achieved drug-free remission after CAR T therapy.
Drug-free remission. Wow, man.
Yeah. For conditions that often require lifelong, debilitating immunosuppressants with severe side effects, these patients were able to stop all their medications and live symptom-free. Some others in the study improved enough to significantly reduce or ditch immunosuppressants altogether.
Incredible.
Beyond this study, other successes have emerged in conditions like myasthenia gravis and stiff person syndrome. This approach truly opens a new chapter in autoimmune disease treatment. It offers a deeper, more comprehensive depletion of B cells, particularly in tissues that traditional treatments often couldn't reach effectively. It provides a level of relief and hope that was previously unimaginable for these chronic conditions.
That's incredibly hopeful for millions suffering from these conditions. Potential remission, not just management. What are the known challenges, though? I mean, you're essentially resetting a patient's immune system. There must be risks involved with such a powerful therapy.
You're absolutely right to ask. There's still much to learn and carefully manage. Scientists are actively researching the frequency and severity of potential side effects, like cytokine release syndrome.
Right, the immune overreaction.
Exactly, which can be serious. They are also meticulously tracking how common and how long-lasting these complete remissions will actually be. The therapy is also complex and expensive, currently requiring specialized centers. So while the initial results are certainly incredibly promising and represent a profound leap, the journey to making it a widely accessible and perfectly safe treatment is definitely still ongoing. It's a powerful weapon, but one that needs very careful handling.
Okay, moving from the microscopic world of cells to the truly cosmic. James Webb Space Telescope, JWST, was science's 2022 breakthrough, dazzling us with its initial images. This year, it continued to astound us, but in a way that's completely reshaped our understanding of the universe's infancy. It's been probing what scientists call the cosmic dawn. What unanticipated discoveries has it made about the really early universe?
The JWST was designed with this incredibly ambitious goal, looking back in time to the universe's first billion years, capturing that faint, red-shifted light from the very first galaxies. However, what it found was a profound surprise, one that genuinely challenged the prevailing cosmological theories.
How so?
It spotted perhaps 1,000 times more galaxy candidates at cosmic dawn than anticipated. 1,000 times. And what's more, some of these nascent galaxies appeared to be Milky Way-sized behemoths.
Huge galaxies that early.
Massive structures that, according to existing models of galaxy formation, simply shouldn't have grown so quickly so early in the universe's history. It was a real cosmic head-scratcher.
So it's not just deepening our understanding, it's actively rewriting it, forcing cosmologists to rethink their models. How are scientists trying to explain these dazzling galaxies that seem to spring up out of nowhere? What are the leading hypotheses?
Indeed, it's a massive puzzle that has really energized the field. Detailed studies this year began offering potential explanations. There are 2 main hypotheses currently being explored, both supported by spectroscopic evidence that's analyzing light to determine composition and velocity.
Okay, hypothesis 1.
One hypothesis suggests that the galaxies weren't actually as physically large as their brilliance initially inferred, but simply incredibly bright. Spectroscopy suggests these early galaxies contained enormous amounts of gas and dust, including heavy elements like carbon and oxygen.
Which shouldn't have been common so early.
Exactly. These elements could only have formed in the bellies of even earlier giant stars that exploded as supernovae, scattering the material. The intense conditions in these early galaxies, fueled by this material, enabled the rapid growth of truly enormous superluminous stars, making the galaxies appear much larger than their actual mass.
Ah, so they were just incredibly bright because of these monster stars. Okay. What's the second hypothesis if it's not just about the stars?
The second hypothesis points to the early universe being rife with active black holes. Spectroscopy in some of these observations suggests massive and active black holes at the centers of these galaxies were a primary source of their brilliance.
Active black holes. Feeding.
Yet not just quiet supermassive black holes. They are actively feeding, drawing in vast amounts of gas, and emitting intense radiation, effectively lighting up their host galaxies from the inside. The mystery this raises, however, is equally profound. How could such massive black holes arise so quickly just a few 100 million years after the Big Bang?
Right, that seems too fast.
Theories now suggest that colossal clumps of material, rather than just the slow accumulation of stars, could have rapidly collapsed under their own immense gravity to form these black hole seeds. It means our understanding of how the universe's earliest structures formed is becoming far more nuanced and dynamically turbulent than we ever imagined. Black holes might have played a much earlier and more active role.
That's truly mind-bending. Black holes as key players right at the start.
Okay, from the vastness of space, let's bring it back down to Earth, specifically to our fields and farms. The next breakthrough involves RNA-based pesticides entering the field. Why is this significant? What kind of problem does it solve that traditional approaches couldn't or did so with unwanted side effects? Traditional insecticides have long been, well, to put it bluntly, blunt weapons. They often work by broadly toxic mechanisms, killing not only the target pests, but also beneficial non-target species. Pollinators, birds, aquatic life.
Yeah, major environmental concerns.
Exactly, disrupting entire ecosystems. The solution approved by the U.S. Environmental Protection Agency, the EPA, this year is called Calantha. It's an RNA-based pesticide spray. And it represents a completely different philosophy. It's designed to target a specific pest, like the Colorado potato beetle, by interfering with a unique, critical gene that only that pest possesses.
So it's much more precise, like a surgical strike, aiming to spare the beneficial insects and wildlife. How does this RNA interference, or RNAi, actually work biologically? It sounds kind of like science fiction.
It sounds futuristic, but RNA interference, RNAi, is actually a natural biological process. It happens for gene regulation and viral defense within all living cells. In this application, when Colorado potato beetle larvae chew on leaves sprayed with Calantha, they ingest these specially engineered RNA molecules. Once inside the beetle, these RNA molecules interfere with the production of a key protein essential for the beetle's survival. The RNA basically acts like a faulty instruction manual, blocking the gene's ability to produce that critical protein, which effectively kills them within days.
And it's specific because the RNA only matches that beetle's gene.
Exactly. The RNA sequence is designed to only match and disrupt the target pest's unique genetic code. We've already seen a genetically modified corn variety on the market since 2023 that uses similar RNA tech to kill corn rootworms. And another RNA pesticide is being developed to target the veromite, a devastating scourge of beehives, promising to protect our crucial pollinators.
That's truly fascinating, a molecular smart bomb for pests. But are there challenges? Can pests, notorious for adapting, evolve resistance to these RNA pesticides like they do with traditional chemicals?
You're right to raise that concern. Pests are indeed notorious for evolving resistance. It's an ongoing arms race in agriculture. For instance, some insects, particularly lepidopteran insects like moths and butterflies, have evolved robust gut enzymes that can effectively destroy RNA molecules. That requires solutions like protective shells or encapsulation for the RNA to be effective.
Countermeasures.
And researchers are definitely concerned about insects evolving resistance to these RNA pesticides if they're not used responsibly, maybe through over-reliance on a single mechanism. This just underscores the need for careful application strategies, rotating different types of pesticides, using integrated pest management approaches to ensure the long-term effectiveness of this powerful new tool. It's not a silver bullet that eliminates all problems, but it's a highly valuable addition to our toolkit for sustainable agriculture.
Next up, an organelle discovery adding a truly astonishing evolutionary twist. The finding of nitroplasts. What's the big revelation here? It sounds like it's fundamentally changing our understanding of biology and potentially agriculture.
This is truly astounding, a genuine jaw-dropper in the biological world. Until this year, it was a long-held biological dogma that no eukaryote, complex-celled organisms like all plants, animals, fungi, algae could fix nitrogen from the atmosphere.
Right, only bacteria could do that.
Nitrogen fixation, that essential process of converting inert atmospheric nitrogen gas into a usable form like ammonia, which is critical for building proteins and DNA. Yeah, is believed to be exclusive to certain types of bacteria and archaea. It was a defining line between simple and complex life.
But that foundational belief changed. A new organelle, a component within a cell, was discovered.
It did. Scientists discovered what they termed nitroplasts. These are unique nitrogen-fixing compartments, remarkably similar in structure and function to chloroplasts or mitochondria, but dedicated to nitrogen fixation. And they were found, surprisingly, within the cells of certain marine algae.
Inside algae cells, wow.
This is a profound evolutionary revelation. It adds a whole new chapter to the story of how complex life arose. DNA studies showed these organelles didn't just appear out of nowhere. They arose about 100 million years ago from a specific, ancient partnership between marine algae and nitrogen-fixing cyanobacteria.
So endosymbiosis again, like mitochondria and chloroplasts.
Exactly. This makes them one of the few known endosymbiotic organelles, following on the footsteps of chloroplasts, which originated from cyanobacteria, and mitochondria, which originated from ancient bacteria. These were once independent microbes that were engulfed by a larger cell and, over eons, became incorporated and indispensable parts of their host organism cells. It's a living example of evolution in action, revealing a completely new pathway for acquiring a fundamental biological capability.
That's a huge deal, linking directly back to how all complex life evolved. What are the future implications of finding nitroplasts, especially for something as critical as global food production?
The implications are immense, particularly for agriculture. This finding, along with ongoing parallel work on diatoms hosting similar nitrogen-fixing bacteria, hint at the possibility of future crops endowed with nitroplasts that would enable them to fertilize themselves.
Self-fertilizing crops.
Imagine crops, corn, wheat, rice that don't need synthetic nitrogen fertilizer. Currently, producing synthetic nitrogen fertilizer via the Haber-Bosch process is incredibly energy-intensive. It's responsible for a significant chunk of global greenhouse gas emissions, and its runoff causes massive environmental pollution like algal blooms in dead zones.
Right, a huge environmental cost.
If we could engineer crops to fix their own nitrogen using nitroplasts, it would be an absolute game changer for environmental sustainability. It would reduce agricultural costs dramatically and fundamentally enhance global food security. It could literally transform how we feed the world, making agriculture far greener and more efficient.
From the intricacies of biology to the fundamental forces of physics, a new type of magnetism has emerged called altermagnets. For nearly a century, physicists knew of only two types of permanent magnets. What is this third type, and how does it challenge our long-held understanding?
For 98 years, since discovering ferromagnets, physicists understood magnetism through two main types of permanent magnets. First, you have ferromagnets, like your iron fridge magnet, where all the electron spins align in the same direction, creating a strong net magnetic field.
Okay.
Second, you have antiferromagnets, where neighboring electron spins point in opposite directions, effectively canceling each other out, resulting in zero overall magnetism. This year, the scientific community celebrated the definitive discovery of a third type, altermagnets. They present this fascinating kind of split personality, combining features of both.
A split personality in quantum magnetism. How does that work? It sounds like a paradox.
It is, in a way, which makes it so intriguing. Altermagnets, like antiferromagnets, have equal numbers of up and down spinning electrons. So zero net magnetism at the macroscopic level. You couldn't pick up a paperclip with an altermagnet.
Okay, so far like an antiferromagnet.
Unlike antiferromagnets, they exhibit these peculiarities in their atomic structure that lead to a very specific, complex asymmetry in their electronic behavior, particularly in what physicists call their Fermi surfaces.
Fermi surfaces, okay.
These are abstract but crucial representations of how electrons move and behave within a material. This structural peculiarity causes them to break time reversal symmetry, a property previously considered the unique hallmark of ferromagnets. So they look like an antiferromagnet on the outside, but they behave like a ferromagnet in terms of their electronic structure and certain quantum properties. It's like having a material that's both magnetically invisible and loudly announcing its electronic presence simultaneously.
So they act like a hybrid, a bridge between the two known types. How is this confirmed experimentally? And what does this new understanding actually mean for future technology?
This wasn't just theory. Multiple independent research groups successfully measured these telltale split Fermi surfaces in materials like manganese telluride and chromium antimonide. That provided definitive experimental confirmation of this new class of magnets.
Okay, so it's real. Why does it matter?
The why it matters here is potentially enormous, because ultramagnets are potentially numerous and combine properties of both magnetic types. They hold immense promise for future technologies. Specifically, they might make ultra-fast magnetic switches in electronics.
Ultra-fast switches.
Imagine a future where your phone or computer isn't just faster, but consumes dramatically less power, extending battery life for days, or enabling entirely new forms of AI processing directly on devices. This isn't just about speed. It's about fundamentally rethinking how we store and process digital information, potentially unlocking innovations we can barely conceive of today, paving the way for truly omnipresent, low-energy smart devices. It's a new frontier in material science and electronic engineering.
Shifting gears from fundamental forces to our planet's deep history, we have a fascinating finding that multicellularity came early for ancient eukaryotes. This sounds like a major paradigm shift in evolutionary biology. What was the previous understanding, and what does this new discovery reveal?
This truly changes a fundamental aspect of evolutionary biology. It rewrites a chapter in the story of complex life on Earth. For a long time, the prevailing belief was that eukaryotes, complex-celled organisms like us, plants, animals, existed as single cells for a staggering billion years after they first appeared.
A billion years solo.
Yeah. The idea was that life spent this incredibly long time just figuring out how to be a single complex cell before finally taking the monumental leap of linking up to form multicellular organisms, paving the way for the diversity we see today.
And what's the new fossil evidence that completely caused this rethink? Where was it found and what did it look like?
Researchers returned to an area in northern China where a similar tantalizing fossil had been found decades ago, but was largely ignored or dismissed as bacterial. Over several years, they meticulously uncovered and analyzed 278 more exquisitely preserved specimens of an organism they named Kingshania magnifica.
Okay.
Under the microscope, these fossils were truly astonishing. They consist of strings of up to 20 cylindrical cells with clearly adjoining cell walls, strikingly resembling modern green algae.
Like little filaments.
Exactly. Furthermore, some specimens even contain distinct spore-like spheres, strongly suggesting specialized reproductive structures within a multicellular organism. Crucial chemical tests confirmed they weren't cyanobacteria, solidifying their eukaryotic nature. This discovery dates multicellularity in eukaryotes to a staggering 1.6 billion years ago, nearly doubling the previous estimate.
1.6 billion. So some of the absolute earliest complex life forms we've ever seen living in intricate communities. What are the broader implications? Does it mean the evolution of complex life is actually faster than we thought?
Not necessarily faster in its entirety, but rather that the initial step towards multicellularity happened much, much earlier than previously assumed. Together with similar recent discoveries from India, Canada, and Australia, these fossils suggest that eukaryotes indeed took an early, profound step toward multicellularity much earlier in Earth's history. However, the path from this simple, filament-like multicellularity to the extreme complexity we see today, everything from jellyfish to redwoods, with specialized tissues, organs, intricate developmental programs, that was far slower. It suggests the initial innovation of cell-to-cell cooperation came early, but the subsequent evolution of truly complex, differentiated multicellular organisms was a gradual, prolonged, and perhaps more challenging evolutionary journey. It teaches us about the surprising flexibility, but also the long timelines of fundamental evolutionary innovations.
From deep time to deep Earth. Our next breakthrough, mantle waves sculpt the continents, challenges previous long-held assumptions about how continents move and are shaped. What was the old theory about plate tectonics, and what's the new one that sounds far more dynamic?
Previously, the prevailing view in geology was that the tearing apart of continents by plate tectonics was a relatively very local process. We imagined hot, buoyant mantle rock rising directly beneath a rift zone, causing the crust to thin volcanic activity.
Right, focus right at the rift.
Exactly. But the old, cold, rigid interiors of continents were believed to remain largely unaffected by these deeper forces, acting almost like passive rafts floating on the mantle. However, groundbreaking new work this year has fundamentally upended that view. It proposes that rifting doesn't just create local upwellings, but actually generates expansive waves in the mantle that actively shape the whole face of continents, even far from plate boundaries.
So not just localized forces, but a deeper, widespread interaction, like a hidden, slow-motion ocean beneath our feet.
It's a fascinating, almost fluid analogy. When rifting occurs, the upwelling mantle material doesn't just go straight up. It rubs against the underside of the cold, dense continental plates above, creating massive, whirling, convective currents of rock.
Wow.
These currents then flow incredibly slowly over millions of years along the continent's underside, like powerful turbulence running under a ship. And as these massive slow-motion waves tumble and churn beneath the continents, they create various profound effects far above, subtly reshaping the landmasses over geological timescales.
What kind of geological phenomena do these mantle waves explain that were previously considered kind of inexplicable?
They explain several long-standing geological mysteries. For instance, they can explain plateaus inexplicably located within the old, cold interiors of continents, far from any active tectonic boundaries. Think of the vast Brazilian highlands or India's Western Ghats.
Oh, okay.
How? The passing mantle waves effectively strip away heavier, dense rock from the bottom of the continental crust, making the remaining crust more buoyant. This causes the continental interior to rise, forming these elevated plateaus.
Interesting.
Furthermore, these mantle waves can stir the mantle in ways that create the right magma mix for kimberlites. Those are incredibly rare and explosive volcanic eruptions that bring diamonds from deep within Earth's mantle to the surface. They can also explain periods of increased erosion on continents, widespread marine extinctions due to changes in ocean basin depth, and even drive earthquake activity in typically quiet plate centers far from active fault lines. It all points to a far more dynamic, interconnected interplay between the continents and the deep mantle than Earth scientists ever thought, fundamentally reshaping our understanding of planetary geology.
That's a truly fundamental shift in how we see our planet working. And finally, a breakthrough that truly captures the imagination, inspires wonder, and heralds a new era of space access that could revolutionize scientific exploration. Starship sticks the landing.
This was an extraordinary technical feat, watched by millions around the globe. SpaceX's Starship, the world's largest and most powerful rocket. It's a colossal, 120-meter-tall stainless steel giant designed to be fully reusable.
Fully reusable.
While it made several test flights this year, the October 13th landing of its immense booster stage was what truly burned into collective memory and captivated the world. After launching the booster, a massive 70-meter-tall structure separated and began falling back to Earth, incredibly faster than the speed of sound.
Faster than sound, falling.
Yet, against all odds, it successfully relit its engines, performed a precise hover maneuver, and was then snatched out of the air by pincering claws on the launch tower it had launched from just 7 minutes earlier. It's a maneuver that truly looks like science fiction brought to life.
That sounds absolutely incredible, like something out of a movie. How does this single feat, this successful catch, herald a new era of affordable heavy-lift rockets and revolutionize space access for science?
The key, and the revolutionary aspect, is reclaiming and rapidly reusing both the booster and eventually the upper stage, which is the Starship itself. SpaceX had already made monumental strides in cutting the price of getting cargo into orbit by about a factor of 10 with its partially reusable Falcon 9 and Falcon Heavy rockets.
Right, which was already huge.
Huge. A fully reusable Starship is expected to slash that price by another order of magnitude, potentially making access to orbit 100 times cheaper than it was just a decade ago. This dramatic reduction in cost fundamentally slashes the cost of doing science in space, democratizing access to the cosmos.
So, beyond just making launches cheaper, what does that truly mean for scientists? The kinds of research they can do? How does it change the very nature of space science?
It means scientists can take more chances with their missions. Instead of needing to design incredibly expensive, hyper-tested, bespoke instruments that cost hundreds of millions and take decades to develop because every kilogram in space is so precious.
Yeah, the cost drives caution.
Exactly. They can now build instruments with cheap, off-the-shelf parts and launch them often. This rapid iteration allows for more experimentation, more data, faster progress. We could envision not one Mars rover, but a herd of them, each dedicated to a specific scientific question, exploring different areas simultaneously.
A herd of rovers.
Or perhaps a fleet of formation-flying mirror segments that could link up in orbit to create telescopes larger and more powerful than anything we can currently imagine. The Falcon 9 already transformed Earth science, allowing companies to launch squadrons of cheap satellites, collecting continuous data, replacing expensive singular behemoths. Starship has the potential to similarly revolutionize deep space exploration. It could lead to the end of incredibly costly projects like the Space Launch System and help NASA boldly go faster, better, and more cheaply than ever before, truly opening up the universe to unprecedented levels of scientific inquiry.
That really opens up the universe, or at least our access to it, in an unprecedented way. Okay, and our final runner-up breakthrough brings us back to our origins, revealing something deeply personal about the human past. Ancient DNA revealing family ties. How has the field of ancient DNA expanded its reach this year, moving beyond broad population movements to something far more intimate?
Ancient DNA analysis has already revolutionized our understanding of deep history. Previously revealing broad population movements, the evolution of diseases like the plague, prehistoric diets. This year, it's gone even further, revealing something incredibly intimate: family secrets. Researchers are now able to precisely reconstruct family trees for people who died thousands of years ago.
Actual family trees from prehistory.
Yeah, connecting individuals not just by shared cultural traits, but by direct genetic lineage.
So we're talking about actual genealogies from the deep past, creating a kind of ancient ancestry.com or 23andMe. How is this level of detailed reconstruction possible? What enabled this leap forward?
It's due to a combination of factors: vastly improved DNA extraction techniques from ancient bones and teeth, and rapidly falling prices for DNA analysis. In the past, studies typically focused on widely scattered individuals to get broad population overviews, like tracking migrations across continents.
Right, big picture stuff.
Now, with an exponential growth in the number of ancient human genomes being sequenced, researchers use something called identical by descent segments of genetic code. These are specific bits of DNA shared between individuals because they were inherited from a recent common ancestor. By analyzing these shared segments, scientists can now estimate relatedness with incredible precision, even out to 6-degree relationships, like second cousins.
Wow, that's close.
New projects are even sequencing hundreds of individuals from a single ancient cemetery, allowing for unprecedented detail.
What kind of fascinating discoveries has this revealed about past societies and their structures? Things we could only guess at before. Can we really see how people lived, how power was transferred?
Absolutely. By meticulously combining the DNA data with rich archaeological information like the age of the skeleton, its burial location, associated grave goods geneticists and archaeologists have been able to reconstruct intricate family trees stretching up to 8 generations deep.
8 generations.
This has revealed incredible, tangible insights into past social structures. For instance, DNA data from elaborate burial mounds of Celtic chieftains in southern Germany showed that 2,500 years ago, power and status were inherited not just through the male line, but often through mothers, suggesting a form of matrilineality.
Interesting.
In stark contrast, kinship analysis of Stone Age European farmers from different sites consistently suggested a strong focus on the male line for inheritance. And moving even further back, we've learned that 2 early modern humans in Europe from 40,000 years ago were part of an extended family, sharing common great-great-grandparents, even though their remains were found hundreds of kilometers apart. It shows level of mobility and family connection in the deep past that we couldn't confirm before.
That's truly bringing the human connections of the distant past into sharper focus. It makes history feel so much more personal, almost like we can see their lives unfold through their genetic legacy.
It does. It adds a whole new dimension to understanding our shared human story, moving beyond just artifacts to the very biological relationships that defined our ancestors. It's a powerful tool for illuminating the social fabric of prehistoric societies.
We've just explored an incredible landscape of human ingenuity, haven't we? From potentially ending an epidemic and curing ancient diseases, protecting our food supply, peering into the cosmic dawn, understanding the very dynamic fabric of our planet. These are truly groundbreaking triumphs that push the boundaries of human health and fundamental understanding. But as we said at the outset, progress is rarely linear. The past year also saw significant challenges and profound failures, serving as a sobering reminder that innovation alone isn't enough. We need foresight, cooperation, collective action.
This is a crucial part of the picture, yes, and it provides essential context to the breakthroughs we've just discussed. We need to acknowledge that alongside these incredible successes, there were profound breakdowns. The first, and perhaps most concerning, is a stark reminder that despite recent global crises, lessons not learned from COVID-19 are still haunting us.
It feels like just yesterday we were living through the painful universal lessons of that pandemic. Millions of deaths, unimaginable economic devastation, stark inequalities in vaccine access that left much of the world behind. The World Health Organization, the WHO, even had an independent panel in 2021 recommending urgent measures shoring up global surveillance, promoting equitable access. But it seems those lessons, born from such pain, may have fallen on deaf ears.
Sadly, you're right. This past year tragically demonstrated a dangerous lack of progress in global pandemic preparedness. For example, the H5N1 avian influenza virus, which virologists have long flagged as a high-priority potential pandemic agent.
H5N1, yeah.
It sparked an unprecedented outbreak in U.S. dairy cattle with confirmed human infections. Critically and alarmingly, the U.S. dragged its feet in collecting and sharing data on the H5N1 spread, drawing worrying comparisons to China's initial problematic early response to COVID-19.
Not good.
Viable virus was even found in unpasteurized milk for sale, highlighting the potential for broader spread. This lack of transparency and proactive data sharing is precisely what fueled the early chaos of COVID-19.
And it wasn't just avian flu. We also saw a concerning resurgence of Mpox, a virus that had previously prompted a global emergency. What happened there? What lessons did that highlight?
Mpox, formerly known as monkeypox, went on a rampage in the Democratic Republic of the Congo and neighboring countries. It surprised the global health community even after a previous global outbreak has supposedly been contained.
Yeah.
This time, the outbreak was particularly devastating for children.
Oh, no.
The DRC, despite being at the epicenter, severely lacked vaccines, and their distribution was painfully slow, especially for vulnerable children. It highlighted how quickly a disease can resurge if systemic issues of access and equitable distribution aren't resolved. And perhaps most concerning for global preparedness as a whole, negotiations on a long-awaited pandemic treaty.
The one meant to fix these issues.
Exactly. Aimed to improve global readiness and ensure equitable responses. Those negotiations hit a dead end this year amid deep and unresolved rifts between rich and poor countries.
So, a crucial global treaty designed to prevent future pandemics and ensure a more coordinated, equitable response is essentially stalled. What were the main sticking points, and what are the implications of that failure?
That's right. It's a significant setback. The core disagreements revolved around issues like equitable access to vaccines and treatments during a pandemic, intellectual property rights for pharmaceutical companies, and the sharing of pathogen samples and genetic sequence data.
The same arguments we saw during COVID.
Pretty much. Developing nations pushed for guaranteed access and benefit sharing, while richer nations and pharmaceutical companies were wary of provisions that could undermine innovation or patent protections. This fundamental rift prevented consensus. While talks will continue in 2025, there's significant worry about the treaty's future, given political shifts and rising nationalism. Helen Clark, former PM of New Zealand, and Ellen Johnson Sirleaf, former president of Liberia, who co-chaired that 2021 WHO independent panel. They starkly concluded this summer that there's been a dangerous lack of progress, warning that political leaders are essentially gambling with our future. It's a sobering reminder that scientific breakthrough alone isn't enough. Political will, trust, global cooperation are absolutely paramount for our collective safety.
And it's not just global health preparedness where we've seen setbacks. Science itself became collateral damage this year due to global conflicts and economic turmoil. How have war and fiscal austerity directly impacted scientific communities and the pursuit of knowledge around the world?
This is a deeply concerning trend. It highlights how geopolitical instability can directly undermine the very engine of human progress. In Russia, due to the ongoing war in Ukraine, officials announced grim plans. Cut civilian research funding by a drastic 25% over the next 2 years, postpone major research facility improvements.
A freeze on science.
Effectively, yes. Employment at state scientific organizations has already fallen by 6%, signaling a brain drain. Meanwhile, in Gaza, the Israeli attacks destroyed the territory's 12 university campuses. This isn't just structural damage, it's the annihilation of entire intellectual ecosystems, impacting generations of students and researchers. And Israeli lawmakers themselves are even proposing a 5% cut to higher education, including research, potentially costing Israel its top global ranking in research spending per capita.
And we also saw significant cuts in Argentina, right? Highlighting how economic instability impacts science even without direct conflict.
Precisely. In Argentina, President Javier Milei's radical economic stabilization efforts, aimed at curbing inflation and government spending, included a devastating 31% cut to research spending. Their main science agency has already lost about 1,000 staff, that's 9% of its workforce, since late 2023. These examples powerfully illustrate how geopolitical instability and economic turmoil, sometimes driven by internal policies, can directly impede the advancement of knowledge, dismantle scientific infrastructure, and lead to a significant loss of human potential and well-being. Cutting off the very source of future breakthroughs we so desperately need. It's a self-inflicted wound for global progress.
Another area that hit a significant snag this year after years of high hopes and promising clinical trials was psychedelic therapy. The drug MDMA, often known as ecstasy, seemed poised for FDA approval as a breakthrough treatment for PTSD. What happened there? Why did this promising new path hit a hurdle?
After decades of rigorous research, MDMA, in combination with psychotherapy, seemed on the verge of a historic moment. FDA approval for severe PTSD. This was seen as a potential watershed moment expected to pave the way for other psychedelics like psilocybin.
Right, opening the door.
However, Lykos Therapeutics' application to the FDA for MDMA-assisted psychotherapy faced substantial concerns from an independent nonprofit and, crucially, from an FDA advisory committee which voted against it. Ultimately, the FDA declined to approve the therapy, asking Lykos to conduct another Phase 3 clinical trial before reconsideration.
That's a huge blow for patients and researchers who had so much hope pinned on it. What were the specific reasons for this setback? Was it about efficacy or something else in the trials?
The primary blame was largely placed on Lykos for flaws in its application and issues with the design and execution of its trials, rather than a definitive statement against MDMA's efficacy itself.
Okay, procedural issues.
Issues like unclear definitions of the psychotherapy component, a lack of key data in their submission, and significant concerns about the blinding of trial participants. In psychotherapy trials, it's notoriously difficult to ensure participants don't guess if they've received the active drug or a placebo, which can bias results.
And MDMA has obvious effects.
Exactly. Its profound effects make blinding almost impossible to maintain, leading to questions about the true placebo effect versus the drug's specific action. So while it's a significant delay, it's probably not a derailment for the field. Some companies are now focusing on the drug's pharmacological efficacy alone, and researchers are actively working to improve blinding methods for future psychedelic trials. This case really highlights the incredibly rigorous and sometimes frustrating path that novel treatments must navigate through regulatory processes, especially when they integrate complex components like psychotherapy with powerful pharmacology.
Finally, let's look at the broader global stage. Environmental negotiations flounder. Despite the ever-growing urgency around climate change, biodiversity loss, plastic pollution, what was the frustrating outcome of critical UN negotiations this year?
Despite the overwhelming scientific consensus and a palpable growing urgency on all those fronts, UN negotiators this year largely delivered little. At COP29, the Critical Climate Conference, rich nations pledged $300 billion annually by 2035 to help developing nations adapt and mitigate.
$300 billion sounds like a lot,
but it falls far short of the $1.3 trillion per year that developing nations say they desperately need and can effectively absorb. This significant disparity drew strong criticism from delegates, who labeled the pledge as paltry and a complete failure, highlighting the persistent lack of financial commitment from developed nations.
So, a significant and disappointing gap in climate funding. What about efforts to protect biodiversity and tackle plastic pollution? Also existential threats.
The news was equally concerning there. At the Convention on Biological Diversity, countries again couldn't agree on sufficient conservation funding, with an estimated $200 billion annually needed to meet global biodiversity targets. Although there was one small bright spot, a new provision was agreed upon, requiring companies that use genetic data from nature, say, in drug discovery, to share revenue with the source nations. A step towards equitable benefit sharing.
Okay, something positive.
But the overall funding gap remains enormous, and negotiations for the world's first agreement to end plastic pollution also failed to make critical progress, specifically failing to cap new plastics production.
Why not?
Primarily due to strong blocking by oil-producing countries and industries that have vested interests in expanding plastic production. As a result, negotiators went home empty-handed, leaving the planet grappling with a worsening plastic crisis with no global regulatory framework in sight.
It's a stark, almost jarring contrast, isn't it? On one hand, humanity achieves these scientific marvels, near total HIV prevention, unlocking secrets of the cosmos, engineering new agricultural tools. But on the other, we see these profound failures in global cooperation and foresight, whether it's learning from pandemics, protecting science from conflict, or addressing existential environmental threats that affect every single one of us.
That duality is precisely what stands out when we look at the year in science and global affairs. The drive for knowledge and innovation, exemplified by something as revolutionary as Lenacapavir or our new understanding of mantle waves, that's a constant. It seems almost inherent to us as humans. We are relentlessly curious, remarkably ingenious, capable of incredible discovery. However, the application and the equitable distribution of that knowledge, our collective ability to bridge that gap between invention and widespread implementation, and our capacity to learn from past mistakes and overcome entrenched interests in short-term political thinking, these remain our greatest and most enduring challenges. It's really a tale of two worlds. Our boundless capacity for discovery met by our often limited capacity for collective action.
And that leaves us with a final provocative thought for you, our listener, to carry forward. Given these dramatic highs and lows of the past year in science and global affairs, this clear demonstration of both our incredible potential and our persistent shortcomings, what specific insight or lesson from this deep dive resonates most strongly with you about the future of human progress? And perhaps even more importantly, what responsibility might that imply for us as informed citizens to help bridge that gap between breathtaking discovery and equitable, effective delivery for all? It's definitely something to mull over until our next deep dive.
This was the Gist Podcast. If you liked it, please leave a like and subscribe to this channel.